Alright, so three types of formed elements. What do we have? Red blood cells, white blood cells, and platelets. Last but not least, let's talk about the platelets. Okay, so again, the cells in your blood, all the formed elements, they come from hematopoietic stem cells. And with platelets, now it takes this path. It goes to myeloid stem cell, megakaryoblast, megakaryocyte, and platelets. So megakaryocyte is, now do I want you to know every single pathway? That's nice to know, and maybe if you take an advanced course, yeah, you probably would have to take that, or if you go med school, you might have to know that as well. But definitely want you to know megakaryocyte, and why is that? It's a very interesting type of cell. So very big, it's a big sounding, complicated sounding word, but when you think of something's mega, right? I think something's like, wow, that's mega important. That's like very important, huge, large. Mega means large. Karyo refers to nucleus. So when you talk about karyon, like things like eukaryote and prokaryote, that refers to the nucleus, right? It feels like eukaryotes, everything that has a true nucleus, whereas a prokaryote, like bacteria, it doesn't have a true nucleus. And then site, again, you should know this definitely by now. Site means cell. So what does megakaryocyte mean? Literally, if you put it together, it's a cell with a large nucleus. And that's why I love these root words, because again, it might be a new word when you put it together, but if you know the roots, you can kind of break it down. Okay, so what about platelets, and what's this megakaryocyte that leads to a platelet? So what we have here is that this megakaryocyte, again, this nucleus is, even though it would actually, if it was to scale, it would be a lot bigger than it shows here. So this is another picture from another textbook, and this whole thing is a megakaryocyte. And these little platelets, these are kind of blebbing off from the membrane of a big megakaryocyte. So again, all this dark purple, this is the big nucleus of that megakaryocyte. But the platelets, these are pinching off, and these platelets actually don't have a nucleus. So they're small little patches of cells that don't really have a nucleus because that nucleus was left in the megakaryocyte. Now, the nucleus of the megakaryocyte, why is it so huge? Why is it so big? Well, the thing is that megakaryoblast, and again, if you have a blast versus a site, blast is immature, site is mature. So when you have the precursor, the younger version of a megakaryocyte, it undergoes DNA replication multiple times, but there's a twist. The cell does not divide. So if you don't know what the cell cycle is and what DNA replication in a cell is, go back to my previous lectures and review chapter 3. I believe that's a cell biology chapter in your book. Alright, so then the cell cycle, there are four main phases. You have your G1 phase, you have your S phase, and this is the phase where a cell duplicates its DNA and duplicates its chromosomes. And then how does it do that? Well, it takes its original DNA, separates the strands, and then if you remember base pairing, and if you don't know what base pairing is, again, go back to my previous lectures. But again, A binds to T, C binds to G. So the thing is that by splitting a DNA molecule, again, you saw that pairing right here, by splitting the DNA helix apart and making a new strand of DNA based on the base pairing, now you have two identical sequences of DNA. You kept the original sequence. Now you just have two copies. Then you go to S phase to G2 phase, and then you enter mitosis. And at the very end of mitosis, this is when you have one cell splitting off into two cells. So a process we call cytokinesis. So again, this is why you need to double your DNA, because if you're taking a cell and dividing it two, if you don't duplicate your DNA, you're dividing your DNA in half and not restoring it. So this is why you duplicate your DNA before you enter mitosis. The thing is that, again, you don't want the cells mutating and changing their DNA every time they divide. So this is why it's very important to make exact copies of the sequence between each daughter cell from original cell. Now, stages of mitosis, again, review it back in chapter 3 if you forgot it. But again, what we have here is duplication, or you already have the DNA duplicated, but then the DNA is being split evenly between daughter cells. Now, with the megakaryocyte, something special happens. Megakaryocytes do not divide. These megakaryocytes, they do not undergo mitosis and cytokinesis during their cell cycle. Or I don't know if they still call it cell cycle in the megakaryocytes. I should look that up. But so what they're doing is basically taking out the M phase, but they're still doing G, and I don't know if they still call it G1 or G2 now that they don't have an M phase. But the megakaryocytes are constantly growing, and they're still undergoing DNA duplication events. So every time they undergo their special version of S phase, they double their DNA, and they grow further, they double their DNA. So they actually do it several times. If they start off with 2 sets of chromosomes, one from each parent, then if they duplicate again, that's 4 sets, then there's 8 sets, there's 16 sets if you duplicate again, then 32, then 64. So the thing is that megakaryocytes, they typically have, like, compared to a normal somatic cell, in other words, something that's not a sperm or an egg, compared to most of the cells in your body, most of the cells in your body have 2 sets, but megakaryocytes, they can have multiple sets of chromosomes due to doing duplication of their chromosomes over and over again, but not dividing it between 2 cells. So why so much DNA? Your DNA contains all the information to make you you, and every single protein, and synthesize every single enzyme and every instruction to make the enzymes that make hormones, all the information to carry out homeostasis in your body and allow your cells to live. It's all encoded in your DNA. Now the thing is that to notice that megakaryocytes are very big, so they actually need to produce a lot of proteins to pinch off these new platelets and also maintain itself. So what does the body need to do to produce polypeptides? Or how do you make a polypeptide? How do you make a protein? You need to make something first before you make a protein, right? All polypeptides are made from or the information to make a polypeptide comes from mRNA, right? But how do you make mRNA? Where does mRNA come from? mRNA comes from making a transcript from DNA, right? So this is, again, going back to the very basics of cell biology. So you have your template. If you have a gene on this is a region on DNA that contains information on how to make a protein. So if you have a gene and transcribe it, you make mRNA. And from an mRNA molecule, you make a polypeptide you use to make a protein. And so from transcription is going from DNA to RNA, and translation is going from RNA to protein.